There are several key challenges. First is the issue of vascularization and maturation. Most current organoids lack of a functional blood vessel system, which limits their size because of core ischemic apoptosis and the ability to mimic real tissue physiology. Without solving this, it's difficult to fully replicate organ level function. I recently saw an article in this year's science paper about the successful vascularization of organoid in several laboratories, but I haven't read it yet. It seems that this problem will be solved in near future. Another major challenge is reproducibility and standardization. There's still a lot of variability between labs in how organoids are generated, which may make it harder to compare results and translate findings into clinical application. Developing standardized protocol is essential. Third, many organoids currently model single organs in isolation, so they often fail to capture physiological complexity and inter-organ interaction. To overcome this, technology like multi-organ system or organoid on a chip will lead to advance significantly. Incorporating the immune system is also a critical next step. Most organoid models lack immune component, which limit their usefulness for studying autoimmune disease, infection, immune therapies. But building in immune interaction would greatly expand their relevance. Scalability is another important issue. To be useful for drug discovery or industrial application, we need to be able to produce organoids at scale and in high throughput, comfortable manner, while maintaining quality and consistency. Addressing these challenges will be critical to unlocking the full potential of organoids in both research and clinical setting.
Okay, so in my opinion, for the next coming 5 years, the most crucial question, also the problem that organoid technology need to settle should be how to proceed from now a fetus level of organ, also the tissue mimicked by the organoids, to an adult, or say more specifically representative of the patient microenvironment of the tissue pathology. So for example, now we use the stem cell based technology to differentiate organoids into a tissue-like structure. Then that usually stays as a fetus level of the tissue. But for the mature differentiation of certain organs that an adult should have, especially for the patients who has the tissue cells to be having the epigenetics with kind of like sickness or the disease type. For that, the current organoid technology cannot really provide a satisfactory solution to represent this kind of disease scenarios. Then we do need to incorporate into some of the epigenetic modifications, maybe mimicking what happens within the patient's tissue cell, especially those of the stem cell within the tissue of the patient. Then we see how to incorporate that into the current organoid technology so that we can really mimic a live patient or at least part of the organ and tissue ex vivo.
Well, I think there are many challenges with the organoid platform. For example, the standardization, because people develop all sort of organoid, and now also including us, we using different type of cells to develop organoid. So it's quite important to compare the organoid developed by different labs using different technologies. How are they similar? Yeah, so the standardization I think is quite essential, and the scalability. Is still pretty difficult to make some organoid, particularly the organoid from normal cells. So they don't grow so well, and compared to some organoid developed from cancer cells. And so if there's is difficulties to obtain large number of organoids, then it will be very hard to do screen type of experiments. Yeah, I think yeah, that's two maybe on top of my mind.
Yeah, thank you.
So that's my work. So, however, there are several reports connecting several organ chips together. Then, but the issue is that whether they could truly succeeded to be resembled to the body. I mean, could truly replacing animal testing or part of a clinical study. And then, but I guess. They just succeeded to culture each cells on a chip and then connect it. So I think it seems to be difficult to mimic the animal body or human body. Then so now we are focusing on the tissue structure. It means having the cellular polarities. Then so it's I believe it be a mimic in the human body. Then so now we are trying to working on liver tissue chip or intestine chip. So I will talk about it later. But so not only to culture the epithelial cells. And also, of course, we have to make the cellular polarity. So it means having apical or basal faces. And also, we are now succeeded to co-culture epithelial cell and intestinal bacteria. Then, so, intestine, so exactly speaking, the capillary blood vessels of our intestine connect to liver via portal vein. And then so drugs delivered into the from the intestine to liver via portal vein. And of course sometimes intestinal bacteria also deliver to our liver. Then, so these drugs and bacteria will meet not at the hepatocyte. Hepatocyte is a parenchymal cells of the liver. So first, maybe so they will meet at the sinusoidal liver sinusoidal endothelial cells. So just culturing the hepatocytes, so it's not enough. So first, I believe the hepatic sinusoidal endothelial cells also very necessary. Then so that's why we are trying to make the liver tissue at least consisting of endothelial, sinusoidal endothelial cells and hepatocytes also. So it's like so maybe it's very important.
Yeah. Okay.
Yeah.
That's excellent question. And reproducibility and comparability are major challenge in the field of organoid research. To address this, I focus on several strategies. First, we standardized our protocol as much as possible, including using defined and quality control culture, media, and reagent. We also rely on well-characterized tissue source, often from a biobank with detailed donor metadata, which help reduce variability. For this, the Korean government controls stem cell banking and distribute it to researchers. Second, we implemented ligos quality control step throughout the organoid development process, including regular imaging, molecular profiling, and the functional assay. Additionally, in the future, we would like to use automation where feasible, such as automated liquid handling system to minimize human-induced variability. But this is too expensive, and unfortunately, there are no plans to equip it yet.
We use many human pluripotent stem cell derived organoid. We mainly use organoid developed using human tissues. An obvious advantage with using that is eventually drug development or treatment strategies will be on humans. So it's quite essential to use human as a cell source, human cells as cell source. And also there are some genetic disease, rare disease that there's no animal models, where the animal models cannot reflect the human pathology. So organoid is probably the only viable platform to study these diseases. Yeah, and it's easy to manipulate compared to animals, and don't cost as much, and easier to go up scale. Yeah, but disadvantage is also obvious. Animal is a whole system. There's for example, you want to study behavior, you cannot use organoid. If the endpoint is behavior, the endpoint of experiment is behavior, then have to test it with animal. So I think the advantage of organoid is to do early stage investigations and do some human specific, ask some human specific questions. At the endpoint, I think is also necessary to test on the animal system.
Organoids offer several compelling advantages over traditional animal models in drug discovery. First, they are derived from human tissue, which means they better recapitulation, human-specific physiology, and disease phenotype. This makes them particularly useful for predicting a human response to drug and for studying patient-specific disease. However, organoids do have limitations. They often lack full tissue complexity, including vasculature, immune component, and systemic interaction that are present in whole organism. Additionally, scalability and standardization across labs can be challenging. That's why I see organoid and animal models as complementary. Organoids are excellent for early stage, high throughput screening and mechanistic study, while animal models remain valuable for evaluating system toxicity, pharmacokinetic, and long-term effect. By interacting both models into the workflow, we can improve translational accuracy and make drug development both more efficient and ethically responsible.
I'm also a young investigator myself. To foresee what may be the important things coming in the next 10 years, or what we want to see of coming in the next 10 years, we think should be really the real applications of organoids in the drug development, which can be widely accepted by the pharmaceutical companies and also government agencies like FDAs. So only with that we will be able to say we are working in a field that really benefit the human life, and also we want to utilize the organoid technology to more or less replace the animal models so that we can reduce the use of animal models. And also by using organoids, we are actually employing the technology that can on our will to closely mimic the patient's conditions. So that may advance the drug development progress and also make us better prepared for the future public health challenges.
Yeah, it's a quite a difficult question. So not a few years. So I have to think about 10 years. So I'm sure I will be able to continue my research 10 years from now. So, but if it's possible, so I told you before. So anyhow, so I will try to create the human body on a chip, or we are connecting all together. And then now we are concentrating on so each organ on the chips. Of course, so it's not just like a single cell also. So we are now anyhow so trying to construct a tissue structure. And then so if I have a 10 years, so anyhow so I would like to create real, not real, but so anyhow so it's really similar or resemble human body, and then to replace the clinic. Of course, not all of them. So part of a clinical study. Yeah. Yeah. Now, so I spent almost 30 years for my study, and then so of course I have to calculate my plan. So 10 years. Yeah.
Well, I think there's still a lot to be done to develop in the organoid system. It's very challenging and it's also very exciting. I think its importance is to use interdisciplinary approach, combine like biology and bioengineering, and also I think the AI approach is also quite necessary, can really empower the whole system. So for young investigators or researchers or students, I think it's an exciting field to join, but be prepared for all the challenges and have a very open mind to always learn new things. Yeah, I think I'm a biologist, a stem cell biologist, development biologist, and I always push myself to learn more tissue engineering techniques and trying to incorporate artificial intelligence, machine learning assisted techniques to empower our own organoid research.
Okay, so for the application for the possible clinical application of organoids for the patient diagnosis or treatment, I think the main problem now to solve will be how to expand and differentiate into organoids from a starting material which is quite small in the volume. So that basically starts from a very small quantity of stem cells. Then we see how to quickly expand them then how to quickly differentiate from the stem cell into the organoids. In order to do that, we need to gain certain insights into the differentiation mechanism of the stem cells with things like growth factors, or say the novel pathways which governs the differentiation of the stem cell into the tissue cells. So that will solve the problem of the fast expansion and the fast differentiation. Once we can find a optimized solution to expand and differentiate, then we can also standardize the protocol that everyone in the world doing the same type of the organoids will use. Then we will be able to quickly expand from a small sampling of the biopsy, then quickly differentiate into a relatively larger quantity of organoids, and also to standardize the application of the organoids so that everyone in different labs working on the same types of organoids, the results are comparable.
Wow, so it's very difficult because honestly, to be honest, I'm a user of organoids, and then so anyhow, so yeah, I'm waiting for anyhow so good organoids. Anyhow, so I would like to request the stable supply. Of course, supply the organoids, and also to request how can I say, same quality on organoids in the world. So not only in one company and one laboratory. So all of them. So maybe yeah, it's very important for scientific research. It's like mouse, so inbred mouse strain. Yeah. So organoids are now also a company supplied, for example, intestinal organoids. But unfortunately, this organoids has one CYP activity. And another big company supplies another intestinal organoids, but unfortunately, these organoids lack of mucin producing cells and so on. So it's not so good. So I'd like to request same quality, means having the same characters. Yeah, it's very important. Yeah.
I think there are two technical bottlenecks for scale-up organoid culture in high-throughput drug screening. One bottleneck is the variability in organoid size, shape, and maturation, which can affect assay consistency. Additionally, handling 3D culture in large quantity demand the price, liquid handling, and the imaging capability that are often not fully optimized for organoid system. The other bottleneck is the cost of reagent and the metrics, for example, matrigel, which are essential but expensive. Automation can help streamline the workflow, but that is very expensive. We don't equip it yet. Therefore, about this question, I answer is I don't know about automation.
Okay. Yeah, I also teach a lot of students in the university. So first, need to really look at your teaching subjects. Are they more experienced, or they are just first year undergrads? I think for maybe undergraduate students is important for them to learn things step by step. First, learn like basics in cell biology, development biology. But for more experienced people want to get into the organoid field to to really consider what they need. Maybe some kind of a modular courses, including like a theory knowledge about different organoids, and it's important to have practice trainings in this, and spend more time on hands-on tryout and troubleshooting, and also provide maybe online resources of most update protocols, and different systems, different organoid systems.
Thank you very much. Thanks.
Okay, so we actually think the individual differences from the tumor organoids originate from different patients is a good thing, because we want to use the tumor organoids to guide the therapy choices from the clinicians. It's because every patient is different, so you can't really find a very standardized treatment. So the advantage of using the tumor organoids is really to represent what actually happens within the patient's body. So that will, at the end, the individual difference. Actually, that's our goal of using the tumor organoids. Then, in order to do that, there are several ways of doing it. For example, we should set up a certain standard protocols that may enhance the replication of the tumor cells while trying to preserve the infiltrated other cell types like immune cells. So with a complex of the infiltrated cells, also the things like endothelial cells, then together with tumor cells, then that can mimic better the microenvironment of the tumors. Then that may provide a better reflection of the patient response to certain drugs.